Welcome. My name is Christopher Schmied. I'm a bioimage analyst at the Fondazione Human Technopole in Milan with Florian Luke. Today, I would like to present the work I did at the FMP in Berlin with developing SynActJ, an easy-to-use automated analysis of synaptic activity. The human brain is comprised of billions of electrically excitable cells, neurons, that communicate with each other to give rise to brain function. At the heart of these communications are synapses, which transmit the electrical information, action potentials from one neuron to the next. This transmission is mediated by chemical neurotransmitters that are synthesized in the presynaptic neuron and stored in the lumen of the synaptic vesicles. When an action potential reaches the synapse, the synaptic vesicles are fusing with the plasma membrane and releasing the neurotransmitters. The neurotransmitters are then able to interact with postsynaptic receptors, which elicit a neurotransmitter-specific response in the postsynaptic cells. Synaptic vesicles are then recycled by endocytosis and refilled with neurotransmitters. To study these highly dynamic processes that occur at the time scale of milliseconds to seconds and to monitor the molecular components that orchestrate them, we use functional imaging tools such as fluorin. Fluorin uses a pH-sensitive GFP that is fused to the luminal side of a synaptic vesicle membrane protein. Under usual physiological conditions, the lumen is slightly acidic, therefore the fluorescence is quenched. When the action potential reaches the synapse, the vesicle fuses with the membrane and exposes the lumen to the neutral pH of the extracellular medium, increasing the fluorescence of the fluorin, as witnessed by the trace. Once the synaptic vesicle is endocytosed, the pH returns to the normal, slightly acidic one, requenching the fluorescence here. Other assays for synaptic function include molecular sensors that detect pre- or post-synaptic influence of calcium, for instance. How does this look like in the flesh? Fast and sensitive wide field live imaging using low light exposure are used to record these highly dynamic processes. Here shown is such a recording where the increase in fluorescence is elicited by electric field stimulation at a specific time frame. We can see that these at single frames and these traces basically that the increase in fluorescence is small in size and intensity around the stimulation. These events are brief elevation of signal relative to the baseline and are very hard to detect by eye, requiring the correct setting of the brightness contrast. Further, we acquire very large fields of view of the live cell tissue culture with many irrelevant structures and autofocus structures also. And around these very small objects of interest here as the blow-up of the insect. The resulting time-lapse movies are therefore hard to analyze, with scientists resulting to a manual analysis or the use of custom scripts that are not widely available to the community, or the use of advanced software toolboxes that require programming expertise or expensive licenses. If we want to eliminate the need for tedious and manual biased analysis, the key challenge is to develop robust and easy-to-use software tools that are widely available to the community. We developed the free and open source feature plugin Synaptic Activity in ImageJ, SynActJ, that allows the fully automatic analysis of synaptic activity in the presented time-lapse movies. Here presented as this workflow, the plugin allows to interactively adjust the parameters on the entire dataset and then the execution of the analysis in batch on this dataset. The plugin can be easily installed using a Fiji update site. How does the workflow work in details? Responding structures are enhanced by dividing maximum intensity projections of frame post-stimulation by before the stimulation. We therefore selectively detect responding structures and removing out non-relevant structures. To improve the detection, we use a Laplacian of Gaussian filter to enhance blob-like structures, and we apply a marker-controlled watershed to produce exact outlines of the responding structures. Further size and circularity filters can be applied. SynActJ enables the robust automatic analysis of responding structures that correspond to active boutons here. We also segment and measure the background of the movie. We further created an RShiny app for the quick and efficient processing of the fluorescent traces that have been extracted by the feature plugin. The Shiny app presents plots for fast quality control, such as counts and areas of the segmented structures, as well as the raw traces and the raw background. The Shiny app then performs basic as well as advanced data processing on the individual movies. The average background value is subtracted from the average signal to compare differences in peak response to the stimulation. The background corrected signal is surface normalized by dividing the signal with the average value of the trace in the frames prior to the stimulation. To compare how fast the signal returns to baseline, a further normalization to the peak of the trace is performed. To benchmark our workflow, we applied SynActJ to an existing dataset that has previously been analyzed manually. Our automatic analysis detects many more regions of interest. We see that 64% of manually selected ROIS overlap with automatic ones. And we looked into what ROIS we were missing with the automatic approach, and these were mostly clusters and large uniform blobs and bright structures, indicating that our automated workflow might reduce manual bias. The cultured hippocampal neurons expressing synapto-pHluorin were treated with DMSO as control, or para-nitro blebistatin. This is a specific inhibitor of myosin 2, and in concurrent with the already published data, we see a decrease in exocytosis after stimulation, witnessed also by the peak signal. We also see that with the automatic analysis approach, we can reproduce the phenotype that the signal is returning much slower to baseline, as witnessed by the endocytosis time constant. When we now compare this automatic analysis directly with the manual analysis, we see that we detect lower peak signal than in the automatic analysis compared to the manual analysis, but particularly in the DMSO control. This is likely attributed to the fact that we detect many more weakly responding events in the automatic analysis. Finally, the variability in the extracted parameters is greatly reduced, with a standard deviation dropping from by a factor of 1.6 to 2.6, which further illustrates the increase in precision of the automated approach. Overall, we were therefore able to demonstrate to you that simple, accessible, and automated bioimage analysis tools can effectively overcome tedious manual analysis, ultimately improving overall the accuracy and precision of the presented analysis. With this, I would like to thank you for allowing me to present this tool to you. I'm happy to answer any questions standing in Gathertown. I want to thank the cellular imaging facility at the FMP with Martin Lehmann, Volker Haucke of the Haucke Lab, and Tolga, Soykan, and Svenja, Bolz as my collaborators, Delia Löwe for technical assistance, Robert Hase for input to the analysis, and Florian Juk at the image analysis facility at HT Milan for hosting.